Thank you so much for this very kind introduction. Indeed, I want to talk about my research topic, which is RNA-Dependent Mutagenesis. And I want to tell you the story how my grandmother made me challenge Darwin. When I was roughly 4 years old, I realized that my grandmother would prefer my sister over me. She got the nicer presents, she got more love and affection. And whenever we got caught up in a fight, my sister was right and I was wrong. And just as 4 years old think, I came to the realization there's just one possible reason for that, because my sister is a girl and I'm a boy. So my conclusion to that problem was, if only I could look like a girl, I would get the same treatment as my sister got. So I asked my mom and my grandmother to sew and to tailor me a dress and a skirt that I could wear to look like a girl. It's needless to say that obviously this did not change my grandmother's behavior, but it is an excellent example that adaptation to environment is everywhere. I want to say it's an instinct. It's something that even a 4-year-old boy can understand, that we have to adapt to different environments. And adaptation is not limited to human behavior. It is a key strategy in survival in nature. It is the reason why we see life in every single corner of planet Earth. So what enables adaptation? One strategy that enables adaptation is the creation of mutations. It's the creation of alterations of the genomic code, the information that holds all the information that we need to live. If only we change this information, we can adapt to changes in our environment. The process that creates mutations is called mutagenesis. And mutagenesis is the reason why we have specification, why we have evolution, why we have brown and black mice, why we have different shapes of flowers, why we have different colors of fishes. So, is there a direction in mutagenesis? Is there something driving mutagenesis? To answer this question, I have to introduce you to two very famous people in my field. One is Jean-Baptiste Lamarck, and the other one is Charles Darwin. Jean-Baptiste Lamarck looked at giraffes and he realized, wow, they have tremendously long necks. And he thought the reason for that is due to the fact that giraffes have to stretch their neck to get to the high-hanging fruits. And over generations and generations of stretching, the neck grows longer and longer and longer. He thought the environment makes the neck longer. It drives the adaptation to this environment. And this happens in an adaptive way. It happens in a directed way. On the contrary, Charles Darwin said, listen, mutations, evolution happens by chance. Mutations happen randomly. It's the environment that assigns a value to each alteration, to each mutation. It makes it either beneficial or deleterious. It's a selection that is put by the environment on the organism that makes it fitter or less fit to a certain environment. He saw that when he studied birds, in particular finches, and he saw that there are different sizes and different forms of the beak, and he found the more crude beak in areas where birds fed on grains, and a more delicate, a more sharp beak in areas where they fed on insects. It's I guess obvious that the hypothesis of Jean-Baptiste Lamarck was dismissed. For centuries we followed Darwin's dogma that mutations happen randomly. But I want to challenge this dogma, not because I think that Darwin is entirely wrong. It's because I think adaptation is such a strong drive in evolution. If adaptation could put a direction in mutagenesis, that would be so beneficial. I want to see if there is not a single true core in what Jean-Baptiste Lamarck said, and maybe the truth lies in between these two hypotheses. Why do I think today is a good day to challenge this dogma that has lasted for almost 200, 250 years? Because nowadays we know so much more about mutations, about DNA, about our chromatin. We have realized that the DNA is not just a single string. It is a three-dimensional molecule that comes in different shapes, in different organizational states, in different packagings. There is an open state where the information is readable, where it's accessible to the cell to read all the information, and there is a more densely packed state of the DNA that is close to condense the very, very long molecule that the DNA resembles. And this is not just a theoretical construct. It's something that we can observe. When we look at electron microscopy images, you see a nucleus is indeed constituted by the brighter open chromatin, the open DNA, and the dark spots are the more densely packed parts of the DNA. And now it becomes interesting, because it was found that when a cell transforms from a normal cell to a cancer cell, in the process that we call chemoresistance, that the closed parts of the chromatin acquire more mutations than the open parts of the DNA. So just by looking at this, we can already say Darwin was not entirely right, that it's completely unbiased. There isn't bias for mutagenesis, a bias for mutation rates in the packaging of the DNA. So are these two states, the open state and the closed state, is this a stable thing? No, it's dynamic. It's regulated by a class of molecules that I study, and these molecules are called small RNAs. You might have heard about the term RNA in the mRNA vaccine for COVID-19. Small RNAs are similar but different. They do not hold the information for an entire protein. They're regulators. And one thing they like to regulate is the packaging of the DNA. They can make open chromatin, open DNA, packaged to a more dense version. And this is sequence specific. This is a class of molecules with all different kinds of sequences. It's a pool that is constituted differently in each cell. And this pool was found to respond to the environment. The environment can change how this pool of small RNA is built up, which sequences are found in this pool. And this pool then can translate into changes in the packaging of the DNA and might ultimately lead to differences in mutation rates. And when we sum up what we see in this picture, in this graph, this is nothing else what Lamarck said. It said that the environment has a direct impact on the mutations and where they happen. So all these individual things are known, but nobody connected the loose ends. And I'm trying to do that because the answer to the question, does adaptive mutagenesis really exist, is of tremendous importance. We've heard by Yara about chemoresistance and persister cells. And when we follow the theory of Darwin, we have to think that resistant clones pre-exist in any tumor and they are selected by the therapy. The therapy makes the mutation that's already there now beneficial. This clone can outgrow the clones that are responsive to the therapy, and in the end the patient is left with a tumor that is fully resistant to the therapy. However, if adaptive mutagenesis is a thing, we can also think of a different model that Yara has also some evidence for, that in the beginning the tumor is not resistant at all, but it acquires the resistance through maybe a state of persister cells, maybe through adaptive mutagenesis, where the therapy itself creates a new environment that makes the tumor cell adapt, that makes the tumor cell create a mutation that is now beneficial in this environment and creates a resistant tumor. In my research, I try to replicate chemoresistance. I try to mimic two different environments, not by looking at the entire genome. Now, I will focus on a single gene. I'll create an environment without small RNAs, and one where the gene that I study is targeted by small RNAs. That should lead to two different states of the chromatin, to an open and to an closed one. Then I will apply some mutagenic agents, for example, radiation or chemical mutagens, and count mutation rates in this gene. And hopefully I will see more mutations when we target the gene by small RNAs than if the gene is not targeted by small RNAs. And that could ultimately give us a clue if adaptive mutagenesis does exist in the model of cancer. So the dispute between Jean-Baptiste Lamarck and Charles Darwin is far from settled. And now there's a third player. This is my grandmother that made me dress like a girl. You can see me here, 4 years old, wearing a skirt together with my sister. I hope you realize the power of adaptation and why it's interesting to study this in the model of cancer. I want to acknowledge my supervisor, Professor Derhavi, and my lab manager, and also the project student, Naomi Green, that worked with me on this fascinating project. And I'm happy to take any questions. Thank you very much.
Yala from Tel Aviv University. Max, beautiful talk. I'm, of course, you're preaching to the choir here. I have a question regarding the gene. Would it be beneficial to choose a gene where if you had an elevated mutation rate, it would increase fitness, right? It's kind of similar to the giraffe. You can think of in mammalian genes that control cell proliferation or anything that would acquire fitness. Do you include selection in your system?
So that is an excellent question. Right now, I'm studying a reporter gene for one specific reason. I want to test my hypothesis in different cell lines and primary cells that have different chromatin states. If I would look at an endogenous gene, it might by default have a different packaging, right, in one cell line to the other cell line. But the packaging is the one thing that I want to modulate, right, through small RNAs. So in the beginning, I will study a reporter gene that I manually introduce into the genome. So I know the exact place, and it's in a genomic safe harbor, as we call it. So I can compare the same gene in 2 different cell lines or even 3 different cell lines. But eventually, this is exactly what I want to do. I want to look at endogenous genes, and I also want to correlate the small RNAs that target an endogenous gene in, for example, a skin cell with the same gene in a liver cell, and see, do I see differences in mutation rates in skin cancer and in liver cancer? That also reflects selection, because the environment for a liver cell is very different than to a skin cell, right? So the ultimate goal is exactly what you were proposing.
Thank you very much for the very interesting talk. I have to admit that I didn't understand everything, but I wanted to ask you a philosophical question, because when you gave the example of yourself with the adaptation, you were conscious, right? You thought about the discrimination, and you thought what would be beneficial. And when you apply it to cells, in order for them to choose the most beneficial adaptation, shouldn't you assume some kind of consciousness? Otherwise, how do they choose the best way?
Yeah, that's a very interesting question. Thank you so much. If I understand you correctly, you ask me if I assume consciousness of cells, right? I'm a biologist. I'm not a philosopher. So I try to answer this question with biology. The thing I don't know if there's consciousness in cells, but I do know that the small RNAs that respond to environments target particular genes, because a cell tries to adapt to a certain environment by downregulating or upregulating certain genes. So they already know how to adapt. They to overcome a different situation. So they already figured that out by their different sets of small RNAs that they can express, and some other sets that they rather don't express in this particular situation. And these small RNAs are already targeting genes. And now I'm asking the question, does this downregulation translate into a mutation eventually? I excluded many details in this talk that are relevant to answer this question. But the knowledge which gene is relevant in what situation is already there, and it is somewhat known in the RNA interference pathway that small RNAs take part in, that certain genes happen to be downregulated. Like you can mimic the same environment 10 times and you will see that 10 times the same small RNA will be downregulated or upregulated. There seems to be some inherent knowledge about what genes might be beneficial to alterate in the expression levels by default. Where does that come from? I have no idea, but this information is already there. It's not a try and error.
Rafael from Bar-Ilan. Do you know if this process can happen through generation, like the generation of the cRNA in the parents, transmitted to the offsprings, and they will probably develop mutation or not?
Excellent question. It happens to be that my supervisor, Dr. Javi, studies transgenerational inheritance of non-genetic information. He studies this in a tiny worm, C. elegans, which is a model organism in biology, and he figured out that indeed there is inheritance of non-genetic information. And this is of tremendous relevance because when we see, and he figured out that small RNAs are indeed inherited in certain organisms. So if small RNAs are inherited, maybe this is an adaptation that is ultimately hardwired by the acquisition of mutations over generations and generations. This is very hard to replicate in humans though, because generation times are quite long in humans. In C. elegans, it's 3 days, right? You can easily create 10 generations, modulate small RNAs in a worm that only lives for 5 days, and study mutation rates. And this is another project that is ongoing in the lab that I study my project in. They do basically the same in C. elegans transgenerationally. I do it in cancer, and they do it in a transgenerational fashion.
Reuven from the Technion over here. So I'm wondering, about kind of the big goal of the project to show how adaptation or how mutation is not random. I mean, there's such a huge difference between an organism on the cellular level than the organism as a whole. And it could be possible that on your cellular level you find some bias for some mutation, but then once you look at the thing as a whole, it ends up being a random. There's so many things going on the way. So how could you really make the connection between some bias on the cellular level to the giraffe really affecting, its mutation on the more macroscopic level?
I don't even try. This is the answer to your question. I don't even try to connect the observation on cellular level to apply to an entire organism because it will fall short. You're totally right about that. But it's important enough to know if it exists in cancer that acts like a population, right? We see evolution in cancer. We see bottleneck phenomena by treatment, for example. We see different branches within cancer development, that certain clones outgrow other clones. So I think if we could answer this question just in cancer, this would be a huge deal by itself, because it would change how we think about therapies, how we think about resistance mechanisms, and ultimately it might lead to a better understanding how we can stop the acquisition of mutations that make cancer resistance, because we have good therapies. It's not that we're lacking good therapies. It's just that eventually most tumors find a way out of the therapy and find a way to become resistant by one way or the other.
Hi, I'm Melissa from Tel Aviv University. I actually wanted to make a connection to Eduardo's talk just before, and I was wondering whether actually studying alien species could help you answer this question of whether mutations are just random or whether there is some boost enhancement from the environment. In particular, like if you look at how quickly an alien species adapts to a new environment, do you actually see that it adapts much faster than what would be expected if mutations are happening like randomly? So is there any data about that? Do you know?
Yeah. So your question was if we if you observe mutation rates in species that are alien to a certain point and compare to the same mutation rate in their home environment, if that could lead to good results, that strengthen my point. I personally have not thought about that because my training is not from an environmental biology side. However, it's a very tempting thought to do that, especially as I told you with the transgenerational aspect that we discussed before. I'm aware of some work that was done in bacteria and also in budding yeast, where they put the same organism in two different environments and indeed found differences in mutation rates that also were targeting certain spots in the genome that were beneficial to overcome this change in environment. But to look at a population level, I did not think about that. But it's a very, very good point.
Thank you. Thank you very much.
Thank you very much.
Thanks, Max.